COVID-19 testing by COBA 6800. The sample is collected here. Each patient is registered with a unique ID. The oropharyngeal swab specimen is collected in viral transport medium. After collection, all the samples are sent to the lab through the pass box. In the lab, the sample BTM is transferred to a level secondary vial inside the biosafety cabinet. After transferring to the secondary vial, it is placed in the blue rack and loaded to the steel tray. Then all the samples can be carried by holding the handle of the steel tray. Here, the amplification plate drawer, where the machine discards the plate after the PCR is done. Next, the drawer number 2 for the solid waste, liquid waste and wash buffer. These are used during the extraction procedure. When it is filled, it should be replaced. As the wash buffer is enough, so we are not changing it. Next, the bulk reagent drawer for the storage of diluent and lysis reagent. The white lane is for diluent and the black lane for the lysis reagent. Next, the consumable drawer for the storage of processing plate, MGP cassette and amplification plate. And finally, the drawer number 1 for the storage of tip racks and the control and the master mix COBA SARS-CoV-2 reagent. We have to simply place the tip racks into the drawer. This is the control cassette. The red one is the positive control and white one is the negative control. We only have to check the sides to keep it correctly. This is the SARS-CoV-2 reagent which contains all the reagents. Now the machine is in the ready mode. simply we have to carry the steel tray by the handle and place it into the input lane of the SSA module. All the sample racks will move through the input lane track. Here is the barcode reader which reads the sample code and arrange it inside the machine. The machine will not start a run until its all prerequisite is fulfilled. With every full batch, 2 clot racks has to be provided. If any clot is detected during the test, the machine will use the clot racks to dispense the tips. Here we can see the sample racks is parked inside the parking platform. As all the samples transferred to the transfer module once job is finished towards the testing. The rest job will be performed by the machine and finally it will give the result whether it is positive or negative. For each batch, 2 tip racks and 2 processing plates are needed. All the samples are transferred to the processing plate in the transfer module. Now we can see the transfer of samples from secondary vial to processing plates. After transferring to the processing plate, the sample rack comes out by the output lane. After the sample transfer, all the samples and other consumables shifts to the processing module where the nucleic acid extraction process takes place. Finally, the purified nucleic acid is eluted to the amplification plate where the master mix reagent is also added. Finally, the amplification plate is sealed and transferred to the analytical module for amplification and detection. We can check the result in the routine control batch section. The whole procedure is about 2 hours 15 minutes. We can take the printout or save it as a PDF. Serological test by Architect I1000SR. The blood sample is collected here. Each patient is enrolled with a unique ID. 2 ml of blood sample is collected by a trained technician. The blood sample is centrifuged at 2500 rpm for 4 minutes to collect the serum. Then the serum specimen is aliquoted to the sample cup. Each patient ID is entered to the machine with proper command of rack position and test selection. The sample rack is inserted into the chemiluminescence immunoassay based automated machine. A single test takes 30 minutes to complete. The test result can be seen in the monitor under result selection.